So I thank the conference organizer for inviting me to this conference and for giving me the opportunity to talk about my recent study. I will discuss three main topics. First, I will give you some backgrounds, such as GIAS and meta-analysis, and I will talk about our new statistical model, RE2C, and with two optimization procedures we applied. And I will show you the performance of our new statistical model. Genome-wide association study has a goal to identify disease causal variants. GIAS tests the association between genotypes and a phenotype, which can be either quantitative or case control status. And we measure the association, measure the amount of the association in terms of p-value. The familiar figure on the bottom of the slide represents the Manhattan plot, of which the x-axis representing the physical position of genetic variants, whereas y-axis representing p-value. The increasing sample size helps a statistical model to find more variants of weak association in GIAS. In this regard, meta-analysis can integrate the multiple GIASes to increase the sample size and therefore boost the statistical power. The best utility of meta-analysis is that it can combine multiple GIASes using only summary statistics. The use of meta-analysis has been extended to integrate GIASes of different genetic backgrounds. For example, it can combine multiple studies of different closely related diseases, cancer types, and different ancestry groups, and even equitars of different tissues. In these cases, a study does not share the same genetic background with other studies, and I want to define the term heterogeneity as the effect size differences between the studies. There are some previous meta-analysis methods, one of which is the fixed effect method to detect a homogeneous effect, and other is the random effect method to detect a heterogeneous effect. Fixed effect method is a conventional meta-analysis framework to detect a homogeneous effect. It can combine the multiple GIASes using only summary statistics of GIASes, such as X as the effect size of each study and V as the variance of each X. In the model, the observation X is the summation of the mean effect mu and the random error term E, where the E follows the normal distribution of the mean 0 and the variance term within study variance term VI. And it tests the homogeneous effect by assuming the hypothesis of no association, and it tests if the mean mu is non-zero. And mu is the homogeneous to all studies in the fixed effect model. To detect the heterogeneous effect, we previously developed Hannan-Askins random effect model, which we call RE2. In the model, the observation follows the normal distribution of the mean mu and the variance, which is the summation of the within study variance V and the between study heterogeneity tau squares. We can build the likelihood function, which contains and not contains mean effect and heterogeneity tau squares. And we can test our model using log likelihood ratio test framework by assuming the null hypothesis of no association for the mean effect mu and tau square heterogeneity. And we test if the mean mu and the tau square is non-zero. Next, I will explain our motivation for our research and two optimization procedure we applied. We found that our first motivation from correlations of statistics. The definition is the use of the same subject in multiple studies that causes the correlations of statistics between studies. And in the figure, you can find that the shared controls between two studies resulted in the correlated statistics. And the sharing of the public controls in multiple studies is now prevalent in the many meta-analyses in GIASes. However, the conventional methods such as fixed effect method and random effect 2 method, which assume the independence of the statistics, cannot account for these correlations. So you shouldn't be confused between the correlation and heterogeneity. Correlation refers to the correlation of statistic, which is the observed statistic between studies, and the source of the correlation is the use of the same subject in multiple studies. By contrast, heterogeneity refers to the differences in the true effect size between studies, and the source of the heterogeneity is differences in the true genetic backgrounds. I found that it is surprising that large consortium data often use the overlapping sample in their studies, and it uses the same subject in the multiple studies, which cause the correlative statistics. The list includes the large consortium data, such as the Psychiatric Genome Consortium, PGC, and WTCC Consortium, and Diagram Consortium. In these cases, correlations can occur in the cross-disease meta-analysis using such large consortium data. The challenge is that there is no meta-analysis framework that accounts for both heterogeneity and correlation. First, RE2. RE2 can account for the heterogeneity, but cannot account for the correlation because it assumes that independency of statistics between the studies. Lin-Sullivan method is the fixed effect method equivalent to the correlated data, so it can account for the correlation, but it cannot detect the heterogeneous effect. Decoupling method is a method can transform the correlated statistics into uncorrelated statistics before we conduct a meta-analysis. So it can account for the correlation if we use the decoupling FE method, but it cannot detect the heterogeneous effect. Of course, we can use the traditional RE2 with decoupling method, but the power of decoupling RE2 is suboptimal. So we proposed our new statistical model R2C and two optimization procedures. First, we developed a meta-analysis framework of correlated statistics, and we account optimally for the correlations. We present our new statistical model, which has the observation is the summation of the mean effect mu and the two random variance components. One of the variance components, which is the U, denotes the random effect reflecting between study heterogeneity, and it contains the tau square. And another variance component, which is the epsilon, denotes the random error, and it contains the variance matrix sigma, which can account for the correlation. Here, the matrix C in the sigma, reflecting the correlation matrix by Lin-Sullivan's method. And we again can build the log likelihood function under the null and alternative hypothesis using the matrix notation we defined above. To test our model with the log likelihood ratio test framework, we have to find the statistics, and in this situation, we must find the maximum likelihood estimator for the unknown parameter in the likelihood function, which is the mean effect mu, and the tau square, which is the heterogeneity. So our proposed model has the log likelihood function with two unknown parameters. Here, we can find the MLEs for two unknown parameters using the simple Newton-Raphson method iteratively to find the two unknown parameters. However, the iterative procedure might be inefficient if we conduct a meta-analysis to test the millions of SNP, which need a lot of computation. To speed up our computation, we applied another strategy to our optimization procedure to accelerate our optimization. Here, we first decomposed our observation X into the two matrix vector multiplication S and Q using direct sum notation. If we use the properties of the direct sum notation, then we can decompose the log likelihood function L1 into two log likelihood function, which of independent observation. Here, L1 prime has the observation Sx and is the restricted maximum likelihood function for L1. We again assume the log likelihood function L1 on the limited parameter space of the mu equals mu hat. In the log likelihood function, transform the log likelihood function. However, the mu hat has to be estimated each time we make a guess for the tau squares, which is in matrix H. So we use the property of the REML and spectral decomposition to make a new log likelihood function, of which transformation reduces the number of unknown parameters into one. Now the parameter estimation needs only the application of a Newton-Raphson method to a single parameter, and the same technique was also used for the population control, population structure control. So we can now build the generalized log likelihood ratio test statistics, which we call R2C statistics. With small modification, we can decompose the R2C statistics into two log likelihood ratio test statistics, one of which is SFE and one of which is S hat. Here the SFE is the same as the fixed effect statistic, which is squared, and S hat has the between study heterogeneity in the studies. Our second optimization procedure is that we developed a new novel statistics to focus on the heterogeneous effect conditioned on the application of fixed effect model. To help you understand our motivation, we first illustrate how we assess the R2C p-value. In the two-dimensional representation of S hat and SFE. When we have observed R2C statistics, then we can calculate the p-value by calculating the probability of SR2C, which is greater than SR2C observed. Now we can map the rejection region of R2C in the two-dimensional representation, SFE and S hat. Here we map the rejection region of two different methods, R2C and FE, and we combine the rejection region into one on the figure on the right side. We found that identified variance by R2C and FE overlap substantially. This can be a problem because R2C is commonly used with fixed effect model. Same as random effect method is commonly used with fixed effect model. Because detecting homogeneous effect is of primary interest in the most meta-analysis injures. Thus, the overlap is bad for the power. We suggest a new statistic, R2C power boost, which focuses on heterogeneous effect. And it has a goal to design a new statistic whose rejection region doesn't overlap with that of fixed effect model. And we have a solution that design a new statistic that is only significant when fixed effect is not significant. Here, we again illustrate how we assess the R2C power boost p-value. First, we can divide the R2C statistics into two regions by using the boundary where the R2C p-value equals to the fixed effect p-value. R2C power boost statistics can be achieved after removing the homogeneous effect, which is dominated by fixed effect statistics. Then there is another problem that there is no simple way to access p-value because the statistic doesn't follow any known asymptotic distribution. So we used approximation for to estimate the p-value of our new statistical model. We first divide the entire range of fixed effect statistics into k small bin and integrated each probability for the small bin to calculate the R2C power boost p-value. This estimation is fast and accurate. Now I will show you the performance of our new statistical model, R2C and R2C power boost. In our simulation scheme, we assume that each SMP has the minor Rayleigh frequency of 0.1 and follow the Hardy-Weinberg equilibrium. And each sample contains 1,000 cases and 1,000 controls. And we assume the meta-analysis to combine 7 studies. In the sampling procedure, we actually simulate the heterogeneity and correlation by following. We first assume the mean effect mu and sample each true effect size using true effect size distribution. And we simulate each observed effect size using multivariate normal distribution, which has the mean of the vector beta and the variance matrix sigma. And the pair of the observed effect sizes are correlated as much as the value of the rho, which we defined previously. Here we test the first positive rate, and we repeated the null simulations of no association 10 to the 9 times, and the first positive rate of our method were well calibrated with or without correlations. Next, we compared the power of the 3 different methods, one of which is the Lin-Sullivan method, which is the fixed effect method equivalent to the correlated data. And 2 other methods, we actually considered the combined use of random effect method with the fixed effect method, as we discussed earlier. So we used the Bonferroni correction to correct multiple testing correction. In the simulation, we considered the 4 different true effect size distribution: normal, uniform, bimodal, and opposite. And we test 3 methods in each true effect size distribution. R2C outperforms other methods greatly in the all scenarios of different true effect size distribution. For example, uniform. In the uniform distribution, DR2 only achieves 24% of power, whereas R2C achieves 71% of power. We fixed the mu so that the power of the best method will achieve 70% of power, and we used the different color scheme for the identified variance by RS and usually found by RE method. And we compared the R2C and R2C power boost method, and R2C power boost method outperforms R2C method in the all scenarios of different true effect size distribution. In the efficiency test, we tested 3 methods, and it shows that R2C power boost can perform a meta-analysis of millions of SNP in an hour. In the real data analysis, we used cross-disease meta-analysis for Alzheimer's and Parkinson's disease by Moskvina et al. We reanalyzed the loci, which is the total 25 loci that are associated with either AD or PD using R2C power boost and 3 other methods, which is Lin-Sullivan, decoupling R2, and R2C. Here the data contains both heterogeneity and correlation, and the source of the heterogeneity is the different genetic background with Alzheimer's disease and Parkinson's disease. And the source of the correlation is the shared controls between 2 studies. In the real data analysis result, we first highlighted the best method, the most significant method for each SMP, and we counted the number. In the result, RS was most significant on 13 SNP out of 25, whereas R2C power boost was most significant in 12 SNP out of 25. And it supports the combined use of a fixed effect model and random effect 2 power boost model. And we found 2 more variants, which is interesting, but which was not identified by RS alone method and became genome-wide significant after we applied combined use of fixed RS and R2C power boost. In summary, previously there was no method that can account for both correlation and heterogeneity. So we developed R2, can account for both. And we further increased the power of R2C by developing R2C power boost. And it shows excellent performances in simulations and shows the utilities in the real data, which contains the heterogeneity and correlations. And this is our website for the software. And I especially want to say thank you to my PI Bomhan and Professor Elijah Eskin in UCLA. And travel funding was generously provided by ISCB. Thank you.
I think we don't have time for questions, so please approach Mr. Lee with your questions later on, and we will. Thank you very much. I should mention that this was one of our proceedings.